Reproduction in flowery plants. The flower. A flower is a bright colored, beautiful part of the plant. Flowers are a reproductive part of plant. Flowering plants are also called angiosperms. Parts of flower. There are different parts of flower, like from outside we will start the sepal, then petals, the stamens, and then the pistil part. First point, sepals. Sepals are the green in color and the outermost whorl of the flower. They are also called calyx. Next is the petals. Petals are next to sepals. They are brightly colored to attract insects for pollination. They are also called corolla. Next is the stamens. It is a male part of the flower. These are the next to corolla. Each stamen is made up of filament and anther. Same is the male part of flower. It is also called endoecium and is the reproductive part of the flower. Next is the carpels. Carpel is a female part of flower. It is the central part of flower. It consists of basal sporon ovary, middle elongated style, and knob-like sticky part stigma. It is also called gynoecium. It is a female part and is also reproductive part of the flower. Functions of flower. It bears male sex organs called stamens and female sex organs called carpels. It is a reproductive part of plant. Ovary matures to form fruit. Hermaphrodite. Flowers that contain both stamens and carpels. Example: pea, hibiscus, and mustard. Next is anther. These are the anthers, the brown colored part of the flower. The structure of anther. The fertile portion of the stamens is called anther. Each anther usually made up of two lobes connected by a connective. Each anther lobe contains two pollen chambers placed longitudinally. All pollen grains of an anther lobe remain ununited in a sac called microsporangia. Structure of microsporangium. Microsporangium consists of two parts. First part is the microsporangium wall, and second is sporogenous tissues. Microsporangial wall. The microsporangium wall are surrounded by four layers. First point is the epidermis. It is the outermost single layer and protective. Second is endothecium. In epidermis there is a single layer radially elongated cells. Cell of endothecium develops fibrous thickening made up of cellulose with the lateral pectin and lining, with help in dehiscence of anther. Middle layer, 3 to 4 layers of thin wall situated just below the endothecium are known as middle layer. Cells of this layer degenerate to provide nourishment to growth, microspore mother cells. Fourth point is the tapetum. This is the innermost layer of the wall. The cells are multinucleate and polyploid. Tapetal cells are nutritive. The tapetum is of two types: amoeboid or invasive tapetum, secretory or glandular tapetum. Functions of tapetum. It provides nourishment to growing sporogenous cells. It secretes hormones as IAA, stored in pollen grains for their early growth. It provides pollen kit covering around the pollen grains. The B part is sporogenous tissues. It fills the interior of microsporangium. Sporogenous cell divides regularly to form diploid microspore mother cells or pollen grain mother cells. Microsporogenesis. The formation of haploid microspores from MMC or pollen mother cells through meiosis is called microsporogenesis. Microspores grow and differentiate into pollen grains as the anthers matures and dehydrate. The wall of the MMC degenerates. The microspores separate and develop into pollen grains. In some cases they remain united in tetrad, for example Juncus typhi. Dehiscence. Mature anther dries up. The tissues between two pollen sacs disintegrates to form single cavity having large number of pollens. Pollen grains. This is a diagram of pollen grain. This is the electron microscopic structure of pollen grain, and this is the post-fertilization, post-pollination event in the pollen grain. It is a male gamete of flower formed in the anther and reach female part carpel through different agencies. The pollen grain is covered by two-layered walls called sporoderm. The two layers are inner antine and outer extine. Intine, the inner layer is made up of cellulose and pectin. Exine, it is the outer layer of pollen made of highly resistant biochemical called sporopollenae. The science of pollen grains is called the palynology. Pollen viability. The period for which pollen grain remain functional is called pollen viability. Pollen allergy. Pollen grains of plants produce several allergy and respiratory disorders. It is seasonal in nature. Some respiratory disorders are asthma, bronchitis, rhinitis. Development of male gametophyte. The development of male gametophyte consists of two stages, pre-pollination stage and post-pollination stage. This is the pre-pollination stage, in which meiosis occur and the separation of pollen grains from the tetrad of 4 haploid pollen cells. Pre-pollination stage. It starts in pollen grains still present in microsporangium. Microspore undergo only 2 mitotic divisions. First division leads to formation of vegetative cell and generative cell. Vegetative is also called tube cell. A temporary callose wall is laid down between two cells. This callose wall dissolves and generative cell lies freely in the cytoplasm of vegetative cell. This is the post-pollination stage. After falling of pollen grains on the stigma, post-pollination changes occur. Pollen absorb water and nutrients of stigma through germ pores. The generative nucleus divides to form 2 male nuclei, which becomes surrounded by cytoplasmic masses and appear as distinct male and male gametes. Pollen tube is formed. The tube nucleus migrates to pollen tubes. Generative cell comes down into pollen tube and divides to form male gametes.